Our next topic is cell transport. On your handout notes, you have a sheet with information. So our notes are going to go here. We have some labeling to do and some color coding. There are two different types of transports, and this is how I have your notes divided up. So we're going to start with this section that is highlighted in yellow, and this section represents passive transport. So transport or particle transport is molecules moving in and out of a cell, which is essential to an organism maintaining homeostasis, which is a stable internal balance. Now you can see in the illustration on this slide that at the beginning, because we can see time is passing, but at the beginning we have all of these little molecules, all of them on one side. And then eventually we start to see them moving over until they're at a state of homeostasis, until they're at equilibrium, or we could say that they're balanced. Now, when it comes to you taking your notes, anything that is highlighted in green is what will go in the note section at the top of your note sheet. Now, normally molecules are going to move from an area of high to low concentration, just as we saw in this last slide. So I'm going to go back here. What we see here at the beginning is what's called a concentration gradient. And what that means is that these little blue molecules, whatever they might be, are highly concentrated on one side compared to the other. So here we see this area is highly concentrated and this area is not highly concentrated. And we see that continuing on until we get to the last part in which they are equal or balanced out. Now, when you see the movement of these little blue molecules from the highly concentrated side where there's so many of them over to the side where there's fewer of them, we saw that they went right across that cell membrane. And what that's called diffusion or simple diffusion. Now, this all falls under the umbrella of passive transport. And these are some key factors to passive transport. You're going with the concentration gradient, which means going from high to low. That's what's normal. That's what's usual, is to go from an area high to a low concentration. So we could say that we're going with the flow. And in both of those situations, we could say that energy is not required. Now, there's three types of passive transport. There's simple diffusion, and that's what we've already seen with the little blue molecules moving from one side of the cell to the other side of the cell, going directly through the cell membrane. Another form of passive transport is called osmosis, and osmosis is just the diffusion of water. And then the last type of transport is called facilitated diffusion. So facilitation means that it's going to be helped in some way. And in this case, it's going to be helped by the aid of a protein that is embedded in the cell membrane. So first, we'll look at simple diffusion again. So in order for these little molecules, and now they're green, but we do see a concentration gradient. We see the high area and the low area. And what's normal is to go from high to low until you fill this space in, or we could say we're balanced, homeostasis or equilibrium. But in order for them to be able to easily move right through this membrane, going right between these little phospholipid molecules to the other side, they have to be small, and they also have to be non-polar, so they're not going to be reactive with water. So on our note sheet, what we're going to do is we're going to label simple diffusion. So that's what we see here in this first picture. So we're actually looking at kind of three separate situations going on this one side. So here we have our low concentration of the molecule, and here we have the high. So we see the high end has 1, 2, 3, 4 of them, and the low just has 1. Now, These are our smaller, our nonpolar molecules, and we are going to represent them with green. So at this point, find yourself a green marker, a green colored pencil, a green crayon, and let's fill in these small molecules with green. Okay, next, water can also move across the semi-permeable membrane from a high concentration to a low concentration. And this occurs through areas called aquaporins. So it's kind of like water pores that are in the cell membrane. Now, when water is moving, we call it osmosis. And you've probably heard the word osmosis before. If you look at the picture on the left, there isn't a concentration gradient, so there isn't any place for the water to move to. And water moves high to low, just like a waterfall. But in this situation, there is. If you see brine water, we're talking about salty water. So now this space is not only filled with water molecules, but it's also filled with salt. So water is going to move from a higher concentration of just plain water to a lower concentration of water. And that's why we see this end going down and this end going up. So now back on your pictures again. Let's go ahead and add in osmosis. So our second little picture here is osmosis. And all of these little guys here, we know what this Mickey Mouse head represents. It represents a water molecule. So there's our oxygen and our two hydrogens, and we're going to color them blue. And if you notice, we are having a concentration gradient. So there's 1, 2, 3, 4 water molecules compared to 2. So we will move across just enough to the other side to balance it out. The last type of diffusion is called facilitated diffusion. So if something is facilitated, you have someone helping. There is help for the situation. And in this case, the helper is a protein. Now, these molecules that are trying to get to the other side, they're too large to move straight through, or they're polar, they're reactive. So there's a couple of ways that this can happen. One is that we can have this protein channel here, so this transport protein, and we've seen these before in your cell membrane notes. They kind of act like a tunnel and allow this big, large molecule to travel through the cell membrane. And then the other way is through a carrier protein. And with this protein, you could see that it's going to basically grab the molecule, it just kind of changes shape and deposits it on the other side. So this one needs help. It needs help because it's too big, and the protein is the helper or facilitator. So now we've got our simple diffusion, no help needed, osmosis, water, and then now for the facilitated diffusion. So we're going to take these larger molecules, we're going to make them red so we can see those little squares. And here is our high concentration. So there we have 1, 2, 3, 4, 5. And we're going to move them onto the other side where there's just one until they're balanced out. So we're not going to move them all over there just so they're balanced out. Now what this is one of those channel proteins, and we're going to color it yellow. This channel protein, as we can see, has this kind of tunnel in the middle of it and acts just like a tunnel through a mountain. So when we look at all of these together, simple diffusion, really small molecules that are nonpolar are going to move from a high concentration to a low concentration. Same thing with water. The water molecules, H2O, are going to move from high to low concentration. And then the larger polar molecules are going to move from high to low concentration. Every single one of these situations, from where we're going from high to low, just to kind of fill in those empty spaces and reach that balance, that equilibrium, because it is so simple and so easy, and it is the easiest pathway to get to equilibrium. All of these situations do not require an input of energy. So basically, they're free for the cell. They don't requare input of ATP.